Hello, my name is Finlay Rollo, and I'm a PhD student at the University of Strathclyde in Glasgow, Scotland. This is our submission for the Radar Challenge 2024, where I will be demonstrating how we extracted micro-Doppler signatures from synthetic aperture radar. Let's begin by discussing what synthetic aperture radar, or SAR, actually is. SAR is a technique that utilizes the motion of a radar platform, typically airborne or spaceborne, to effectively synthesize a longer antenna. This process increases the spatial resolution of the radar beyond what would be possible with the physical antenna length alone. By moving the platform over a target area, it is possible to generate high-resolution images, such as the SAR image of Denver you see on the right-hand side. Now let's talk about micro-Doppler effects. These effects occur when parts of the radar target have their own velocity in addition to the main bulk target movement. The classical example is the rotating blades of a helicopter. These induce an extra frequency modulation, the so-called micro-Doppler signature, on the radar returns, with an example shown in the time frequency plot below the SAR image. This is helpful because unique target movements can create distinct patterns, helping us analyze and identify complex targets. The main aim of our project was to investigate if these micro-Doppler signatures were detectable and extractable in a lab-based SAR system using the phaser kit. The motivation behind this is twofold. Firstly, to develop a practical understanding and hands-on experience with SAR and micro-Doppler phenomena. Secondly, to create a lab-based setup that mimics the capabilities of a much larger and more expensive system that would typically be used in a commercial setting. This avoids the high costs associated with such setups. Moving on to the methodology of our project, let me walk you through the setup and the experimental procedures we used to obtain our data. Our experimental setup includes a 2-meter-long linear axis rail, which is capable of moving the phaser kit along its length with a maximum speed of 0.45 meters per second. This rail allows us to simulate the movement of a radar in a controlled environment, allowing us to create our synthetic aperture. As shown in the image on the left, the rail is equipped with a small motor, which ensures precise control of the speed and position of the phaser along its length. The target used in our experiment, displayed on the right, is a simple metronome covered with aluminium foil to enhance its radar reflectivity. In terms of the radar waveform, we utilized a burst of 1000 ramped pulses, each pulse having a pulse repetition interval, or PRI, of 2 milliseconds. These pulses were organized into a slow time, fast time matrix to allow us to conduct our post-processing and focusing. The radar system operated at a bandwidth of 450 MHz, affording us a range resolution of just over 30 centimeters. The basic micro-Doppler analysis was conducted using spectrograms of the cross-range samples obtained after range compression. This analysis allowed us to visualize the frequency modulations caused by the small movements of the swinging metronome arm. Through this method, we were able to replicate real-world SAR processing in a lab environment. That was an example data capture from one of our experiments, and in this segment I'll explain the pipeline we used for processing the captured radar data. The first step in our data processing pipeline is range compression, with range compressed data shown in the first image to the right of the setup video. Range compression was achieved by using the range response function in MATLAB. The phaser kit was set to G-sharp on receive, and so the FFT method was specified. From here, we are primarily interested in the micro-Doppler signature of the target. We can extract this directly by taking a spectrogram of the range bin containing the modulating target. In this case, it is the 7th range bin down from the top. This visualization shows how the frequency changes over time, and we can see fairly clearly the sinusoidal pattern caused by the moving metronome arm. These signatures will be further analysed in the following slide. Following range compression, we can also perform azimuth compression to create a focused image of the scene and as depicted in the second image. A basic range Doppler algorithm was used in which an azimuth FFT was used to transform the data to the range Doppler domain. Range-dependent reference strips were created for each range bin, and focusing was achieved through convolution of these reference strips and the cross-range samples. As the synthetic aperture used in this experiment was very small, the effects of range cell migration, which occurs when the distance between a target and the radar varies over the acquisition, were ignored. This pipeline demonstrates the capabilities of our miniature system in a controlled lab environment, allowing us to closely simulate and study micro-Doppler signatures. Now to showcase some results. This slide presents the results of two different acquisitions, one when the metronome was not moving, or a stationary target, and the other when the metronome arm was moving, or a vibrating target. On the upper left side of the slide, you can see the focus SAR image of the stationary target, and the image shows the target's position. Below this, the spectrogram of range bin 7 illustrates the lack of any significant Doppler shifts, which is consistent with a stationary target. Moving to the right side, we have the results of a vibrating target. The focus SAR image shows a similar response, however there is an apparent yet expected shift in azimuth in the vibrating target's image due to its non-zero velocity and consequently Doppler. This shift demonstrates the phaser's capability to differentiate between stationary and slowly moving targets. The real point of interest is the lower right spectrogram of range bin 7 of the vibrating target, where we see a fairly clear micro-Doppler signature. This signature matches well with the ground truth, where the metronome was set to 70 beats per minute, or a frequency of 1.16 Hz. And this has a corresponding period of 0.86 seconds, which matches fairly well with the spectrogram. The spectrogram captures the periodic motion of the metronome, highlighting that it is actually possible for the miniature SAR system to detect and visualize micro-Doppler effects due to moving targets. These results further validate the methodology presented. Before we conclude, let's discuss some limitations we encountered during our project. Understanding these limitations is crucial for future work and improvements. Firstly, our setup was limited by the number of samples we could capture per frame, and we only had 2 seconds of truly continuous pulses. We attempted a stop-and-go method in which we could take multiple readings and increase the signal-to-noise ratio. However, we lost coherency between the frames, and the out-of-phase terms significantly increased the clutter in the images, as shown in the example image on the right. Additionally, conducting experiments in an indoor environment introduced challenges such as multipathing. Multipathing occurs when transmitted signals reflect off various surfaces before reaching the radar receiver, creating echoes and additional noise in the data. Lastly, we faced synchronization issues between the rail system and the radar acquisitions. This affected the timing and alignment of our collection, impacting the quality of our results. Addressing these limitations in future iterations of this project will be important for enhancing the performance of the setup. To conclude, this project demonstrated the feasibility of using a miniature SAR setup with the phaser kit to extract micro-Doppler information. Despite the encountered limitations, we successfully captured and analysed the micro-Doppler signatures of vibrating targets. The results aligned well with our ground truth, highlighting the capability of our setup. Moving forward, addressing the limitations such as multipathing and synchronization will be our focus. Improvements in these areas will enhance the system's accuracy and applicability in further lab-based experiments.